You start your class.
Right.
Good morning, all.
Morning, sir.
Morning. Go back to mute. Go back to mute. Just want to know whether you can hear me. So let everybody mute yourself. Then again, all of you that join using your phone name, change the phone name to your own name. You know, this is for the purpose of attendance. If you did not change it and you have problem later on, you cannot tell me you attended the class because your name is not there. It's iPhone or Techno or Samsung that attended the class and not you in particular. So change the name of your phone to your name. I'm saying this now so that there will be no problem thereafter. So change your name, change your phone name to your real name now. I can still see Somebody with 2525, maybe that's the matric number. Change your phone name to your real name. And in the chat box, write your full name and your department and your matric number. It's very, very important. If you don't do that, just pray that you don't have problem later on. You will not be attended to. Simple instructions, some of you still fail to follow it. As I'm still speaking, I can still see some. Two 525030323. That just maybe matric number. Include your name there. Include your name. Change your phone name to your own name and your matric number. And in the chat, write your full name, your department, and your matric number. You can hear me now. I won't say more than that. Good morning once again. So let's start the class right away. Can you see my slide that I'm sharing with you?
No, sir.
Can you see it now?
Yes, sir.
Okay. So we stopped at homozygous and heterozygous the last time we met. So today we are continuing with more of the key terms in genetics. So we are starting today's on with hemizygous. What do you mean by hemizygous? Hemizygous is a condition when genes are present only in one copy. Hemi means the gene present only in one copy. Genes on X chromosome in male have one X and one Y chromosome. Whereas genes on Y chromosome, only one Y chromosome in males. In females, we have two X, but in males, we have only one copy, only one X and only one Y. If you disturb my class, I will not hesitate in sending you out. I don't know why it is so difficult for some of you to follow simple instruction. Don't speak until I ask you to speak. I have muted all of you. Some of you are still unmuting themselves. I don't want to hear your voice. You can't be disturbing my class. Some of you join the class while you are in the motor park, and I can hear the conductor shouting his destination. Some of you join in the market. Some of you are in the bedroom. That is not even my problem, but don't disturb the class. Mute yourself. What's the problem about that? So when gene is present only in one copy, then you said it is hemizygous. For instance, in male has X and Y, and female has XX. So if only one is present, especially in some disorders, for instance, in Turner syndrome, if female has X naught instead of XX, it's a condition in female. So that individual is said to be hemizygous because only one copy is present. The other copy is missing. So that is hemizygous. Don't confuse hemizygous. Don't confuse hemizygous with homozygous. In homozygous, there are two copies present, only that the two copies are the same, are identical. That's where you have XX, as in human. That's why you have ZZ as in birds. So the two are identical. But in hemizygous, it's only one that is present. So instead of XX for homozygous, in hemizygous, it should be X0, X0, X0. So it's a condition in female. Okay, it is called Turner syndrome. The female possess such a female has some characteristics. Okay, it's equally a disorder. So that's hemizygous and homozygous. Then in dominance, dominance is the ability of an allele to express itself phenotypically, both in homozygous and in heterozygous. Now you can see homozygous. Here we use capital TT. Okay, so if an ability or a trait or a character is expressed when the two alleles are identical, capital now, that is dominant, TT. Okay, that's homozygous. And in heterozygous, even when it is one capital and one small letter that is present, which we call heterozygous. You know, we have discussed heterozygous to mean one capital and one small. That is, the two are not identical. Don't look at capital T and small t as being the same T, only that they occur in different cases. Fine, but they are not identical. In genetics, you don't say they are identical because one is capital and the other one is small case. So meaning that they are heterozygous. Then in recessiveness, it is the ability or the inability of an allele to manifest its phenotype in heterozygous. So you can see in heterozygous, only capital T will be expressed. The small t will not be expressed because it is recessive. So small t will only be expressed when the other allele is equally small t. So when a recessive allele is present with a dominant allele, it will not be expressed. That makes it recessive. So we equally go on to hybridization and hybrid. So hybridization is the process of crossing two genetically different individuals. Crossing two genetically different individuals. As you have it, we have the homozygous tall, that's capital TT, versus homozygous dwarf, which is small tt. Okay, and when you cross the two, then there will be production of gametes. So the homozygous tall will only produce gametes that are capital T, and the homozygous dwarf will only produce gametes that are small t. So when you now cross the two gametes, you can see this is what happened during fertilization to produce zygote. The zygote that is produced is capital T and small t, which is now heterozygous, So the heterozygous zygote that is produced is now said to be the hybrid because it's the progeny of hybridization. That is the progeny of crossing two genetically different individuals is called hybrid. So when you hybridize, what you have as the product will be called hybrid. Then we equally tall of monohybrid. Monohybrid simply means crossing involving one pair of allele at a time. Crossing involving one pair of allele at a locus under study. So when you want to study the inheritance of a trait, you want to see how that trait is being passed from one generation to the other. It's being passed from parents to offspring. And you talk of monohybrid. Mono simply means one. You are following the inheritance or the transmission of one trait at a time. Then when you talk about dihybrid, dihybrid is simply means the crossing or the study of the transmission of two pairs of allele at two different loci. Two pairs of allele, okay, at two loci under study. So di, from the word di means two, mono means one, di, two. You can equally have trihybrid. That is three pairs of allele. You can equally have tetrahybrid. That is four pairs of allele, and so on and so forth. So when you now cross single, one pair of allele, okay? When you cross one pair of allele between two individual organisms, then you have monohybrid cross. Monohybrid cross. Then when you cross two pairs of allele, we have dihybrid cross. Then we talk about generations. We have F1 and F2. F1 simply means first filia. Okay? F means filia. Filia meaning son. So when you talk about first filia, that is the son directly from a parent. When you talked about second filia, that would be the grand. Okay, it may not necessarily be son. Let's just say child. Child. Somebody cannot be disturbing us here. I've warned you enough. I'll send him out. Adesuyi, I will send you out. I said mute yourself. What's your problem with Oju Elegba? I have done the needful to him or her. I've removed him from this class. And Mohammed, Khadija, remove your video. Remove your video. So when you talked about first filia, that will be the children directly from a parent. When you talked about second filia, then those one will be grandchildren from the same parent. Okay, you can have first filia, second filia, so it's like children, grandchildren. Then you can equally have third filia, and so on. That's great grandchildren. You can those are the meaning of filia. So when you now have F1, F1 simply means first generation. That is progeny of a hybridization of crossing between a male and a female. Then we have F2, which are the hybrid from F1. When you now cross to F1, you have F2. That is when the children of F1, when they marry, they have their own F2. So in extending this to ourselves, you as an individual, you are the F1 of your own parent. You are the F1 of your own parent. So when you have your own children, okay, your own children will be F2 to your parent, but they will be F1 to yourself. So that's filial generation. So here we cross a homozygous dominant parent with homozygous recessive parent. So the homozygous dominant parent will produce all the gametes, that is capital T. The heterozygous parent will produce all gametes that will be small t. So when they cross, then we have F1 that will be heterozygous. So when you now cross two of F1, or even when you cross the F1 with another individual, okay, maybe with recessive parent now, okay, you now have the gamete. This heterozygous individual will produce two types of gamete. Some of the gamete will be capital T and some will be small t. But this individual that is recessive will only produce one type of gamete. All will be small t. So when you cross them in their F2, you are going to have some of the offspring that will be capital TT. Oh, sorry, there will be no capital TT. Some of the offspring that will be capital T and small t, and some of the offspring that will be small tt because of the random combination here. So now, when you cross, remember, we cross F1, which is heterozygous of two parents that are homozygous. You know, these two parents are homozygous. One is homozygous dominant, one is homozygous recessive. When we cross the two, we have F1 that are heterozygous. When you now cross such a heterozygous offspring with an individual that is homozygous recessive, just like one of the parents here, okay, what we are doing is called back cross. Okay, we are doing back cross, okay, because we want to know, okay, this is test cross, not back cross, because we want to know the actual zygosity of the parent, whether homozygous or heterozygous. But here that we cross, we now have at their own F2, half heterozygous and half homozygous. You know, the homozygous here will be recessive. So if we have such a thing, that tells us the zygosity of this parent is homo. When we have half-half, I will tell you more of this as we are moving on. I just want to fast forward it because it's still part of what we are going to see. So the next one is reciprocal cross. Reciprocal cross. I want you to get it very well. Two reverse crosses in which the sexes of the parents are interchanged. Okay? The crosses. When you carry out a reciprocal cross, it means you made two separate crosses by reversing the sexes of the parent. What do we mean by that? Look at this first cross. We cross a tall with a dwarf individual. But that tall is having this symbol. This symbol represents a male, and this symbol represents a female. The symbol you saw, you can see in front of tall is representing me. So we cross a tall male with a dwarf female. That's one. When you now do a reciprocal cross, it means you now change the sexes based on the trait. So you now change the sex of the male, okay? That is, you want to do another cross in which a male will be dwarf with a female that will be tall. So when you see the reversal, okay, reversal or interchange that you are seeing here, we interchange. We didn't convert a tall male, okay, to a dwarf female. So so after this, we now look for, we will get some results in their F1, okay? Then we now look for a male that is dwarf and cross it with a female that is tall. Now, we get some result as well in their F1. We are trying to do something. When we do this, we try to compare whether the result will be the same or different. So if the traits, okay, that is the traits we are considering here, you know, we are considering tallness and dwarfness. If the traits are autosomal, the reciprocal cross always yield same result because it is autosomal. The traits are being born by both male and female in equal proportion. So there will be no difference in this. But if the traits are sex linked, okay, if the traits are on sex chromosome, remember I told you earlier on in the last lecture that we have some traits that are autosomes, and we have traits that are on sex chromosomes. So if the traits in the male and female, we are interchanging their sexes, is on sex chromosome, the reciprocal cross will give a different result. You know, if the traits are autosomal, the reciprocal cross will always give us same result. But if the sex is on sex, if the trait is on sex chromosome, we are not going to get the same results when we carry out a reciprocal cross. Then we have what is called back cross. Back cross. Back cross is the cross of F1 offspring with one of its parents. With one of its parents. So the back cross or test cross that I mentioned earlier is a way, is a type of back cross. It's a type of back cross. You know, we cross the F1 with one of the parent. In this case, the parent that we cross with is the homozygous recessive. So it's a back cross. It's a type of back cross. But back cross, okay, can be carried out with either of the parent, whether it is homozygous dominant parent or a homozygous recessive parent. So that is back cross. But I said test cross is a back cross. When that back cross is done with the homozygous recessive parent, then it is called test cross. You can now see the difference between the back cross and test cross. Okay, it's not really a difference, but a form of, so back cross, okay, involve test cross. So a test cross is part of or is a type of back cross. You know, back cross simply means crossing the F1 with any of the parents. Okay, but when that back cross is done with the recessive homozygous parent, it is called test cross. All right. So now look at what I have here. If F1 is crossed with dominant parent, all progenies, F2 will be dominant. All progeny will be dominant. Okay, because you know, we are crossing with a dominant parent. So all the offspring will express the dominant trait. If it's crossed with recessive parent, individual with both phenotype will appear in equal proportion. That is, if back cross is now done with a recessive parent, so we are going to have phenotype of the two parents in equal proportion. That is, we are going to have those that will be tall, and we are going to have those that will be dwarf, as we have it here. You can see when this is done with this, what we have is this and this. They are all in equal proportion. Ratio 1:1. So the ratio of progeny produced during back cross is called back cross ratio. Like I've said now, ratio 1:1 when it is crossed with dominant parent. When it is crossed with recessive parent, we are going to have equal proportion. That's ratio 1:1. But when it is done with dominant parent, okay, we are going to have every of the F2 being dominant. So we may not have the ratio in the same proportion because one will be missing. Now, getting to test cross. I don't need to waste more time here because I've explained it, but just for the benefits of those that were not listening there, test cross is a type of back cross. I said it, in which the F1 progeny is crossed with its double recessive parent. That is the recessive parent. And test cross is used to determine whether the individual exhibiting dominant character or homozygous or heterozygous. So test cross I said it is done to determine the zygosity of the parent or the zygosity of the F1 offspring. So if you don't know because when we cross with, when that was done and we got all of them to be dominant, all F1 are dominant. So we may not know we may not know whether all those dominant ones are recessive heterozygous, okay, or homozygous dominant. So to now know that, we carry out test cross. So if we now carry out the test cross and all of them are, dominant, that shows the parent, okay, that result at F1. They are all homozygous. But if they are heterozygous, okay, all the offspring at F2 will now have equal proportion. So that's that. So you can see when you cross homozygous dominant and homozygous recessive at F1, we have all of them to be dominant. So suppose we saw this phenotypically, we didn't know the genotype. So how do we now determine whether F1 are homozygous dominant or heterozygous? So we now cross it with the recessive parent. When we cross it with the recessive parent, which we did here, and we got F2 to be of the same proportion, then our F1 will be heterozygous. But if we got our F2 for all of them to be dominant, then that means our F1 are also homozygous. So these are some of the key terms in genetics. Okay, so let's go on to the work of Mendel. We mentioned Mendel as the father of genetics, and I told you he carried out his work on garden pea, which is Pisum sativum. Why did he choose Pisum sativum? Because offspring produced by this plant can be quickly and easily produced. So you can quickly replant them and get your result again. So he cross-fertilized pea plants that had clearly opposite characters. For instance, tall with short, smooth with wrinkle, those containing green seeds and those containing yellow seed. That is what we call 7 pairs of contrasting character. So Mendel noticed in this garden pea 7 pairs of contrasting character in the same species. He noticed that some of them are tall and some of them are, dwarf. Some produce smooth seed, while some produce wrinkled seed. So he noticed about 7 of that. And then, why was Mendel successful in his work with Pisum sativum? Okay, number 1, the plant is very easy to cultivate in open ground. It's a legume. So when you cultivate it in open ground, it quickly grows and produce. And then the flowers are normally self-fertilized. So self-fertilization takes place in the flower, so you don't need to carry out cross-fertilization. And the plant shows contrasting character. That's 7 pairs that we are talking about. And when a contrasting character is crossed between 2 individuals, that's monohybrid cross, then the hybrid are perfectly fertile. So the hybrid are fertile, and that they can also be taken into the next generation. James Sunday, I'm going to remove you if you don't close your video now. So, and then the plants are normally self-fertilized, as I've said. But if you want to do cross-fertilization, it's also not difficult. So it can be done easily. Then that is when you allow the plant to cross-fertilize, maybe by wind, but you can do it yourself. That is artificial fertilization. You can transfer the pollen grain, okay, from one plant to the stigma, to the stigma of the other. That is, you can artificially fertilize the plant. So that's what he did. And Mendel did not compound the problem by wanting to study the transmission or the inheritance of all the 7 characters at a time. That will lead to confusion. So what he was doing is monitoring the inheritance of one character at a time. And while he was doing this, he was maintaining statistical records. He was recording his observations, which is very, very important. And these are the 7 pairs of contrasting characters that he noticed on the Pisum plant. And then, not just noticing, after carrying out his work, the inheritance of each of them at a time, not at the same time, because it's going to give us more, it's going to be difficult. And, you know, at that time, for him to do that will make it tedious and maybe, you know, overwhelmed by the kind of results you get. So it was just monitoring the transmission of a trait at a time, not minding what is happening to other traits. So, and on that 7 traits, he noticed that after his work, his carefulness on the work, he was able to distinguish between the pairs, which one is dominant, which one is recessive. So the length of the stem, so tallness is dominant over dwarfness. The position of the flower. These are the characters, and these are the expression he noticed on those characters. So when he was considering the length of the stem, he found out that some are tall and some show dwarfness. But he found out that tallness is dominant over dwarfness. So also for the remaining characters, position of flower, color of the pod, shape of the pod, shape of the seed, color of the seed, and the color of the cotyledon. So because he carried the inheritance one after the other, so he was able to know which expression is dominant over the other and which one is recessive. Excuse me. This is another illustration of the 7 traits that Mendel studied. So all these now, after he studied, okay, culminated in the propounding of his laws. That is Mendel's laws of inheritance. Mendel's laws of inheritance. So after all the work on the Pisum sativum, so he came up with these 3 laws. Actually, there are 3 laws, but because, you know, the scientific method, before a law becomes a law, it has to pass through stages. You know, hypothesis, you know, statement or hypothesis. It is when that hypothesis is right or valid that you take it to a theory. Okay. So some stopped at theory. They couldn't become law because there are some, you know, what do you put it now? There are some exceptions. There are some exceptions that will not make such a theory to become law. So they stop at theory. So to become law, there must be no exception at theory, and then it becomes law. then that makes it valid. So out of the three, only two were able to scale through to become laws. That's why we always talk about the two Mendelian law. So the first law is the law of segregation, okay, which states that during gamete formation, during meiosis, the alleles for each gene segregate. That is, they separate from each other so that each gamete carries only one allele for each gene. So that's law of segregation. The two alleles in the gene during gamete formation will separate so that a gamete will only have a member of an allele. Then the law of independent assortment. That one to say that genes for different traits can segregate independently during the formation of gametes. You know, the first law is the law of segregation, which is talking about a gene containing two alleles, and the two alleles separate. The second law, which is independent assortment, is equally saying that as one gene is segregating in the law of segregation, because it's not just one gene, other genes too are equally segregating in such a way that the segregation of a gene does not affect the segregation of another gene. So they segregate independently, they assort independently. So that is the second law. Then he equally talked about the law of dominance, though that one did not become a law. What he said was some alleles are dominant while others are recessive. Okay, so an organism with at least one dominant allele will display the effect of the dominant allele. This one is true to some extent, but not totally true. Okay, some alleles are dominant while others are recessive. Fine, it's true, but not totally true. Why? There are instances where alleles, okay, will neither be dominant nor recessive. There are instances where a gene will contain alleles that one is not dominant and the other is not recessive. Okay, we talked about codominance. You have heard that before. Okay, we equally talked about incomplete dominance. So the law cannot be 100% true, but to an extent, it is true. Okay, in the same sense that we have some alleles that are dominant and we have some that are recessive in a gene. In a gene, okay, an allele must be recessive, an allele must be dominant. That is according to Mendelian inheritance. Okay, but you know, more work took place after Mendelian, and they found out that in a gene, the two alleles in a gene may be codominant. Okay, and at times the two alleles in a gene may undergo incomplete dominance. Then we have a situation of multiple alleles. Multiple alleles, that's a gene will have more than two alleles. So when they have more than two alleles, so after taking dominant and recessive, what do you call the other one? So those are the exceptions to the third law. And that is why this law is not always found, because it has limitations, unlike the first and second. So let's quickly see how Mendel was able to illustrate the two laws. The first law, which is law of segregation, he illustrated it by using monohybrid cross. Now we know what a monohybrid cross is all about. So that is the cross between two individuals, okay, using a character, mono, one trait. So parents differ by a single trait. So that's what you do. crossing two pea plants that differ in pod color. So the trait is pod color of the pod of the fruit of flagging. So you notice that some pods are green and some are yellow. So you now cross two pea plants, one having green pod and the other having yellow pod. So you represent the green pod by capital GG and represent the yellow pod with small gg. Remember, I've told you earlier on in the table that green pod yellow pod. And that's why he used that. So the green is capital GG for green and small gg for yellow G. And the capital GG with yellow G got all the F1 offspring, all of them green. All F1 offspring are green because green color is dominant over yellow. But that tells you, you remember, green is capital GG, yellow is small gg. That means all the offspring that are green, they are what? They are heterozygous. They are heterozygous. So F1 generation is heterozygous. And then they will now, all the green here will now be capital G, small g. Capital G, small g. So as we have it here, so this is equally doing the, so it now went to F2. You know, it took 2 of F1. Remember we said these F1 are capital G, small g. It now crossed 2 of them. That's in F2 generation. So when it crossed the 2 green F1, which are capital G, small g, it got what we have in this table. So the genotype it got is 1 capital GG, which is green. Then we have 2, okay, that are equally green, but they are heterozygous, capital G, small g. And we have the last one to be yellow, which is small gg. You know, when you cross this, look at this. This is what we call, what is the diagram now? Okay, so when you cross this, okay, capital G and small g, as you have. So this is G, this is capital G, this is capital G. You have this. Then when you cross this with capital G, you have this. Then when you come down, you cross this with this, you have this. When you cross small g with small g, we have that. Okay, so that gives us the genotype. But the phenotype and the genotype, the ratio is 1 capital GG to 2 capital G, small g to 1 small gg, which is ratio 1 to 2 to 1. Okay, in phenotype, when you are looking at the phenotype, 3 of them are green. That's phenotype. Remember, I defined phenotype for you at the beginning, that it is the express trait from the genotype. So express physically, both physically and physiologically. So we can see 3 green and 1 yellow. So phenotypic ratio will be 3 to 1. 3 to 1. So the second law too, which was based on the results of the first law, okay, it now went ahead by illustrating it using a dihybrid cross. That is, using 2 traits, the inheritance of 2 traits at a time. And what did he do? He crossed a green, okay, a green pod containing yellow seeds with yellow pod containing green seeds. There are 2 different traits. For the pod to be green does not mean what will be inside will also be green. So it can be green, it can be yellow. And for the pod to be yellow does not mean the seed inside must be yellow, because they are two different trees. So, but what Mendel did was to cross a green pod. You know, that's what we did to illustrate the first law. We cross green pod with yellow pod. But here, he now cross a pod that is green, but the seeds inside are yellow, with a yellow pod with seeds inside to be green. Okay, he crossed the two. And you know, at F1, because he crossed through green and through seed. When you look at the table again, yellow seed is dominant over green seed. Don't be confused. Once again, green pod is dominant over yellow pod. But when you come to seed inside, yellow seed is dominant over green seed. So you now cross a dominant green pod containing dominant yellow seed with a recessive yellow pod containing recessive green seed. So when that happens, what do we have at F1? All the F1 offspring contain green pod, because that's the color that is dominant for pod, and yellow seeds, because that is the color that is dominant for all the seeds. So that is why he used this genotype, capital GG, capital YY, cross with small gg and small yy. Okay, to have our F1 offspring. The F1 offspring will now be capital G small g, capital Y small y. So he proceeded to F2. Okay, he proceeded. So this is the result of that's the result of proceeding to F1. So where he crossed two of these, G, G, Y, Y. I mean, capital G, small g, capital Y, small y, which is the genotype of F1 offspring. So he crossed the two of them. But we have to know, how do we have the gametes that will be produced? So for this individual with this genotype, there are 4 possible gametes that the individual can produce. How do you mean? You know, some of the gametes, remember our segregation and independent assortment. So as this allele, capital G, small g, as they are segregating, capital Y, small y too are equally segregating. So you can have in one allele, capital G with capital Y. In another allele, in another gamete, we can have capital G, small y. In another one, we can have small g, capital Y. And the last possible one is small g, small y. When you combine the two alleles, these are the 4 possible gametes that could be produced. So when you now cross 2 of these, that means one with this possible gametes with another one with the same possible gamete. That's what happens in this in the table here. This table here. So what do we have? Can you see it clearly now? Because I've enlarged it so that it will be easily visible. So you now have on top here one of them, and here you have another one. So when you now cross this with this, this with that, and everything, you are going to have 16 different genotypes. 16 different genotypes. Because when you look at the genotype, no 2 genotypes are the same. So there are 16 different genotypes. But when you look at phenotype, there will be 4 possible phenotypes. 4 possible phenotypes, which are, you have some of these 16 that will have green pod and yellow seed. Green pod, yellow seed. When you look at the table and you count them, you see there are 9. For instance, 1, 2, 3, 4. That's the first row. All the 4 there, they have green pod, yellow seed. That's 4. When you come down, that's 5. That's 6. Okay. Come down again. You see 7, 8, and lastly 9. I mean, yeah, 9. So they have green pod, yellow seed. Then another one will be green pod, green seed. That's green. When you look at them, that's 1, 2, 3. Then when you look, the next one, that's yellow pod, yellow seed. That's yellow. How many? That's 1 here. That's 2 here. And that's 3. Then the last one, we have yellow pod, green seed. That is the 2 recessive, which is only 1. That's it here. So these are the possible combinations. So the F2 genotypes, okay, and phenotypes are here. So those that have green pod, yellow seed, these are their genotypes. There are 9 of them, according to what we did in the table, as we saw it in the table. Okay, and then for the green pod and green seed, they have this genotype. So also this, and then the last one, they have that. So they are these. We have 9 genotypes and we have 4 phenotypes. 9 genotypes and 4 phenotypes. So when you look at the table, okay, you see 5 of them that have, 9 of them have the same genotype, okay, as in yellow, green pod, yellow seed. Those that have this, when you count them, they are 9. Okay, and then we have those that have green pod and green seed, that's having this. So when you look at this, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9 possible genotypes. So for those that have green pod and yellow seeds, they will have this. You have this when you look at them. And those that have this phenotype, they have this genotype. So that's how, but as I said earlier, you will see that even the 9 possible genotypes corresponding to 4 phenotypes, they don't have the same genotype. This and this are not the same. This is homozygous for the 2. This is homozygous for 1, heterozygous for the 2. And likewise, all others. So you hardly see any of them that is having. But because we have 4 possible genotypes based on the ratio 9 to 3 to 3 to 1. So we have that. Now, according to Mendel, okay, remember we are talking of Mendel's inheritance now, Mendel's genetics. There are some assumptions that Mendel's assumed as at the time he was carrying out his work. He assumed that all genes have 2 allelic forms. Okay, that is, any gene, I'm not ready to entertain any question now. So, for Larry, I will call you when I'm done. All genes have 2 allelic forms. So that was his belief. Okay, then one allele completely dominates the other. One must be dominant and the other recessive. You know, this is the 3rd law that I said is not, it didn't scale through. Then, equally said all traits are monogenic. That is, any trait that is being expressed is are being responsible for by one gene. That is, one gene, one trait. All these are assumptions, but we now find out that they are not true. A gene can contain more than 2 allelic forms. That's multiple allele. Then we now find out that one allele, not in all cases, that we have one allele completely dominating the other. That is one dominant, one recessive. No, we have cases of co-dominant. Then all traits are monogenic. No, it's not one trait, one gene. We found out that a trait can be under the influence of many genes. Okay, and vice versa. Influenced by a gene as well. So all chromosomes occur in homologous pair. Okay, those are his assumptions. All genes assort independently. No, not all genes assort independently. We have linked genes. So you will get to know this in advanced genetics. And we found out that a mutation in a single gene causes a disease in all cases. Okay, but there are some that equally follow this. And that's why we cannot rule out all these assumptions totally, only that they are not 100%. There are cases where these assumptions are applicable, but not in all cases. These are assumptions of Mendel, which is law. So let me run this class up with the importance of Mendelian law, importance of the law. So let me go straight to that slide. Can you see what I'm sharing? Hello?
Yes, sir, we can see it.
Yes, sir, we can see.
Is anybody there?
Yes, sir.
Yes, sir,
Yes,
we can
sir.
see, sir.
So I said, I know the time is fast spent. So let me quickly go to the last one. So I suppose to take you through some exceptions to Mendelian assumptions, where we have codominant, where we have incomplete dominance, where we have multiple alleles. So those should be, okay, should be ringing in your mind, though I didn't go into details, but exceptions to Mendelian assumptions are found in cases of codominance, cases of incomplete dominance, and cases of multiple alleles. Multiple alleles. So get to know that and read up those three. They are very, very important. You may have 1 or 2 questions coming out in depth. So don't say you are not taught. I am telling you to read those 3 cases up now, because you are going to have, you may have questions coming out from them. Once again,
Please, can you say it again?
once again, okay, let me do it this way. Let me just put it in a slide so that you can see it. Okay. So these are exceptions to Mendelian assumptions. Can you see the three? Can you see the three now?
Yes,
Yes,
sir, we
sir.
can see it.
Yes, sir.
I see the
Yes,
Yeah.
sir. Yes, sir.
Yes, sir.
Codominant, incomplete dominant, and multiple alleles. So these three did not follow Mendelian assumption. Like I said, those assumptions are valid to an extent, but not in all cases. So exceptions are to that assumption are these three. So lastly, where and how can genetics, the knowledge of genetics could be applied? For those of you that are studying medicine, you can see genetics has application in medicine. So also in agriculture, all the GMO food that we see these days, okay, they are based on the knowledge of genetics. So you can play with genes. You can edit genes to suit your purpose. So you can edit by insertion and by deleting. So, and to suit your purpose. Then in medicine, okay, disorders that we talked about, genetic disorders, okay, in vaccine production, all those use the knowledge of genetics. Then in criminology and forensics, so you can use genetics to apprehend a criminal, to investigate a crime. So also in evolution, we ought to know that as a result, evolution is based on, inheritance of traits and passing it down, okay? So, and at times, mutation can occur, which can also lead to evolution. So all these are applications of genetics. You can read more about their details, but for the purpose of this Bio 101, I don't need to go into too much details for this. So with this, I say thanks you for listening. And this will be the last time I'm having my class with you. But we'll be meeting in the lab and also during the exam. So if you have any question, now, I'm here to answer you. Any question? Hello, no question? So if there's no question, I'm going to close the class for today. Who has a question? You don't know how to raise up your hand to be recognized. Mohammed Khadija, do you have any question? Kudirat Uluwatobiloba, yes, your hand is up. I'm listening.
Yes, sir. I'm not actually asking a question. I also asked if we will send the slides.
Well, I don't normally give students my slide. And that's why you have everything to yourself. Many of you are taking screenshots of the slide. So what again do you want? I can send the record of this class to you. Because if you have the slide, do you know the explanation that goes with it? So when you are listening to, the class, the record of the class as well as the slide, you tend to gain more. Is that okay?
Yes, sir.
All right. There was one David raising up hands the other time. Is he still there? Or he has gone?
Yes, sir. I'm here. I just wanted to confirm about the slide.
Okay.
I've already heard. Thank you, sir.
Okay. You're welcome. Khadija, are you raising up hands or which one are you doing? You can see this is what I was saying. I said, change your phone number to your name. Who is this iPhone that is raising up hand? Is iPhone going to talk now? If you can, you see, I pray you don't have issue with this class. So if I check through the record and I find that you that is using iPhone is having the issue, you're on your own. No. What is so difficult about changing the name of your phone? Who want to see the type of phone you are using? Does that matter to anybody? So, any other question? Any other question? Mohammed Khadija, can't you hear me? Unmute and talk.
Yes, sir.
You are raising up
Good
your
morning.
hand. Good morning.
No. Okay.
I said you should close your video. You are showing your video. Everybody is seeing you.
Okay.
Okay. All
Okay,
right.
sir.
So another class will take place on Wednesday, but not by me, but by another lecturer. So until then, have a nice day until we meet again in the lab this evening for some of you, but definitely not all of you will come today. So see you then.
Thank you, sir.
You're welcome.
Thank you,
Okay,
sir.
sir. Thank you, sir. Bye, sir.
Bye.
Extreme sick.